Okay, welcome everybody. Hello. This is the KBase Educators Fall 2025 orientation call. So this is kind of a welcome to the KBase Educators program. We are going to walk through what exactly the Educators program is, so a bit of background. Then we're going to go through an overview of KBase Educators resources, our landing page, just how everything is organized. We'll start to actually get into some of the teaching narratives and have some time for practicing along with demo. We'll also have some space for breakout groups so we can break into different groups and do some work together. And then we'll also go through any of the newer resources and newer kind of like released items. So to get started, because I know in my little survey that you filled out when you're registering, we have about 2/3 of the folks who signed, or 1/3 of the folks who signed up are completely new to KBase and haven't used it before. And then about half of folks either haven't used KBase or it's been a while since they've been on the platform. So our goal is to really get you nice and oriented with kind of KBase itself, what the platform is, and then get you into the educators org and give you some time to practice. So there's my little overview, and we'll also provide some updates for the program. My first question, and you can put this into chat just so I can get the know about who's here present right now. Can you let me know if you use computational biology, bioinformatics, or data analysis in your courses right now? Okay, we have the whole gamut of not yet, but planning to. So hopefully we'll give you some resources so you can get started during this presentation and introduction. I know I don't have to explain to you why a computational approach can be useful or helpful with students, but just for some context, when you are implementing more computational biology, you can do a lot of different things, especially with microbes. So you can see the genes that are within that organism and have your students think about what those gene products might do. You can also compare and contrast functional capabilities from those known sequences when you're going through assembling and annotating genomes. You can also build phylogenetic trees. So that's inferring the evolutionary relationships between organisms, either within a community that you've sampled, and then also just the identifying who is there. So especially with those unculturable microbes, then we can identify what different species might be present. And one way to do that is to use KBase. So I'll give a little introduction to the Department of Energy Systems Biology knowledge base itself, which is the platform that we use with the KBase Educators program. So KBase itself, it's a community-driven research platform for systems biology. It's really designed for bench biologists and also works really well for instructors. The goal is to synthesize data into finding insights and transforming complex data into understand more of these environments and these places that we're exploring. So the platform allows users to access public data as well as uploading their own data. And most of these are data will be provided from projects that are funded by the Department of Energy, and also allow access to open software tools and the compute power so that you can design your own analysis and really customize your workflows. So as I mentioned, KBase is funded by the Department of Energy, specifically the Biological and Environmental Research Program. This program sponsors different user facilities and resources. This includes the BER Structural Biology Resources, Joint Genome Institute. This would be a user facility for sequencing and advanced genomic research. EMSL, which is the Environmental Molecular Sciences Laboratory. That's also a user facility that provides a lot of cool equipment for different folks to use. And then we can also think about behind data curation and publication, the National Microbiome Data Collaborative, which is an expert group in microbiome metadata standards. They also provide some support with standardized workflows for microbiome data. ESS Dive, which is the environmental system science data infrastructure for a virtual ecosystem. This is a data repository for earth and environmental sciences. And then also KBase. And so KBase is kind of where we're coming in for the data analysis, visualization, and also supporting publication of data analysis behind computational biology. And we can think about KBase coming into this whole research scheme behind, you have your collecting and processing your samples, and then KBase will come in that analysis and visualization stage. So KBase itself democratizes access to sophisticated analyses. We're really thinking about the pillars of FAIR, so findable, accessible, interoperable, and reusable through this free and open access platform, where we enable folks to integrate their own data, access public data, as well as accessing tools and creating their own customized analyses, sharing with collaborators, and then publishing their results. And when we think about kind of how these all fit in to a classroom or courses, we can think about these customized analyses and how they're shareable and really building on what we call the narrative. Those are our analysis workflows. And within the KBase Educators program, we've kind of leveraged this particular system to build resources. And these are really resources that have been developed by peers, so fellow educators and the community, based on kind of what I've been identified as the most important needs, things that we're wanting to address immediately. So really community prioritized resources that were also designed in an iterative and educator design process. So we did a lot of build, test, learn with our feedback, and they pull from the KBase capabilities of the behind using the narrative. So being able to integrate data that's already available, tools for teaching workflows with the students, and that free compute power. And so the different resources that we have developed are primarily related to the narrative, which are teaching workflows in KBase. So we've gone through and we've created some different workflows that are modularized. These workflows are adaptable, so they're customizable, they have flexible content, and they're also shareable. And we have 6 different workflows that are available with data already loaded. These include genomics, metagenomics, phylogenetics, metabolic modeling, pangenomes, and transcriptomics. So these are all broken down across a few narratives, and we'll look at the narrative architecture and how all of these different workflows are broken down. But essentially we have like genomics, for example. There's one workflow that's associated with assembly, and then you can import your data into the next workflow for annotation, and then from there going through more genome analysis and genomic comparison. Likewise with metagenomics, we go through the whole data import stage, then into assembly, read-based taxonomy for metagenomic data, binning, bin-based taxonomy, and then more evolution exploration from there. And so these are just kind of the blueprint and examples of resources that are already available to use, but can be customized. And we'll dig into more of how you can go through and customize these different workflows themselves. All right, so that, and then I can also share just a few anonymous student feedback. So we have gotten some great feedback from students, both in just courses that do data analysis, as well as course-based undergraduate research experiences, where students felt like they were really getting to do real-life experiments and data analysis, and being able to use a platform that they felt was acting like scientists. And I really like the last one in the blue quote, which the student said that they felt like they were able to do such a complex looking work in a relatively short amount of time. And they were impressed by how many computer programs and algorithms exist. And they thought they just scratched the surface. So for those who've not yet to see the narrative itself and how we've gone through, and if we think about it, building out these different workflows themselves, we are working in what we call the narrative. So this is where you're building your different analysis workflows. This is where we have all of the data, the different tools, enabling the ability to put in commentary through the markdown cells. This is really scaffolding where you can add in more outlines and information for students. There is ability to code as well. And all of these narratives provide persistent access and are reproducible. So these can be shared with colleagues, with students. You can also edit them as you create different versions for different classes with different data sets. And the idea behind these different resources and making them available for educators is that the community of everyone who uses these, they're able to join the educators community and the KBase Educators org, which will get you in if you have yet to join that. The idea is that you can access these resources, using them to teach, adapt them, so using them for your students with the data that you're interested in, and then also sharing ideas and telling us how we can improve these different resources, contributing your own to the community and sharing them with others, and how we share these different, for the most part, narratives, but you're also welcome to share other resources beyond the tools used in KBase, and we'll look at a couple of those, are through organizations, and organizations is part of the infrastructure of KBase. So these are essentially a space where educators can join them. So it is a space where we can share data and narratives among different users. This is a great way to also set up a method for your classes to share narratives or the different teaching workflows with your students all in one space. And you can manage the members, whether it's accepting them to join the org or removing them from the org, as well as associating different narratives themselves. We have the KBase Educators org itself. Lana has shared the link to the KBase Educators org. So if you have yet to join, please go and request, and we'll get you into the org, and we'll do some exploration of the different teaching narratives that we can see here. But this is essentially just a landing spot within KBase where you can access all the different narratives and and I suggest to just log into KBase, and if you have an account, if you don't, we'll get you set up with one. But it's a bit easier than going through the search, which is an option. You can search for them. You just have to make sure you toggle to all organizations. And so who belongs to this community, who has access? We have over 270 members in the KBase Educators org right now, and they are across about 27 different countries, which was the last count. And we have a lot of different institutions, 238 different institutions, and they include a lot of different students that we also support. If we think about our 54 different minority serving institutions, as well as 100 emerging research institutions, which is our current count of different organizations. So it's quite a few different academic communities that are represented with and have access to all these teaching resources and are contributing as well. So for the educators organization, for those who are on, does anybody not have an account for KBase? And if you don't, you can show a hand. Yeah, okay. Everybody has an account. That is awesome. If not, when we stop recording, we can have I can help you out and make sure you have an account with KBase. Again, you'll want to go to the link that Lana shared. And if you can share that one more time, Lana, for the access to the KBase Educators org, then you can access that. If you have yet to set up a KBase account, we have some really great documentation, and this is also within our virtual notes document. So in case you haven't joined the virtual notes document, we have the links to where to sign up for KBase. We have a quick start guide for some information on how to get started. And we also have a nice outline. If you go down to the workflow and activities list, here's where we also have a link to the educators org that you can join. And Jacqueline, just to answer your question, do we need to form an organization ourselves for your students to share a specific protocol? You do not have to start an org, but it really depends on how you set up your class. So we'll talk more about best practices when we go through our KBase educators handbook that we have. Okay, so what I'd like to do next is just a nice walkthrough of our different resources that I've mentioned following our flow, and then we'll go into a bit more on KBase itself. So different resources we have are the KBase Educators landing page. So this is external to KBase itself. This is a place where you could find a lot of different resources. Another spot where we have the educators org. We also have an educator handbook to the educators org. We have a YouTube playlist. This has a lot of our previous fall orientations are recorded there. We also have a general, much deeper intro to KBase that's accessible there. We have links to blog posts about our different working groups and how we've gone through to develop resources. We have a link to MICROnet, which I will talk about at the end with our updates, as well as our microbiology resource announcements collection. So this is a great place to access some of the different resources. So you can go directly to the KBase Educators org from here. You can also get to the educators handbook, which is a PDF. We have a PDF version of the handbook. This has a lot of different resources in it as well. There is information on how to join the KBase Educators organization, also how to join our user Slack and the educators group there. We have a code of conduct, the different resources. So we've gone through again kind of the links to getting started, what to do if you need help with KBase, accessing our YouTube channel, our documentation, the educators page. And then we also get into best practices. So we'll talk a bit more about teaching logistics with creating orgs, copying and sharing narratives, setting up your course. And then there's more information on contributing as a community member. So if you want to share your own resources with the community. Let's see, I'm going to do a quick pause on the recording. Alrighty, so when you are using KBase and using the narratives, and again, that's what we call our analysis workflows. This is where we are working in KBase. You have a few different options. So you, for uploading your own data or using data that's already available, this is really just we're going to do a walkthrough on the different capabilities and orient some with terminology behind using the narrative. Again, that's our name for the platform that we're working in KBase before we log in and look at the narrative ourselves. So for data itself, you can upload and import it securely, either using drag and drop method. This is really for data that is about 2 gigabases. Gigabases are smaller in size. If you have much larger data, then you'd want to use your Globus endpoint. And you can import a lot of different data types. This includes sequencing reads, so FASTA, FASTQ files, assemblies, isolate genomes, so already annotated genomes, metagenome assembled genomes, metabolic models, expression data, and more. The platform itself will have suggested import parameters based on file type detection. But again, you might need to select particularly what type of files. For example, in the screen grab that's shown here, they're FASTQ reads, but you have to determine whether they are interleaved or non-interleaved based on the information that you have from the sequencer that you used, if it's your own data, or the metadata that's already been associated with that file if you're importing from SFA. You can also add multiple files and data types at the same time when you're doing your upload. This is enabled for a few different types of data. And if you have really a lot of data sets or a lot of data that you need to import at once, we do have a bulk import option, and we are happy to support you and direct you to figure out or to find our documentation and help set up a manifest for that. Once you have your data in the system, there are a lot of different apps that you can then analyze your data with, and we mostly categorize these by function. That includes read processing, genome assembly, annotation, comparative genomics, sequence analysis, again, metabolic modeling, both isolate and community specific tools, viral tools, and then we also have some tools based on utility. So if you are working with multiple data sets or need to do any bulk items, that's where you're going to go. And then we have the app catalog itself that you can access, and we'll take a look at that when we get into the narrative. And then I mentioned sharing workflows. So you can share them with different individuals or with organizations. Inherently, the narratives are private. So when you first create a workflow or a space that you're analyzing your analysis in, this is going to only be accessible by you. But then you can share these particular workflows through the managed sharing, either with individuals or with groups. So that's through our managed sharing button. And this really allows for a lot of different collaboration at different levels. And then also, if you are ready to publish and share your workflow with others, you can make your workflow public. When you make it public, people only have the view option. So if you're sharing with individuals, that's where you can give them the edit, save, share option. But unlike working in a Google Doc, you won't be able to simultaneously work in the same narrative at once. So if somebody's working in a narrative on one computer, it will show that it updated to a person who might have it open on another computer and has edit access. So that's the one caveat, especially for students who might want to work on them at the same time. They'll just need to have one computer that they're working at together. And then again, for publishing itself, you are able to generate static narratives, which are essential versioned HTML snapshots of a narrative that is accessible outside of KBase. It's really nice. It shows all the different data as well as the citations from all the tools that you use within that particular workflow. And it can also, we can get a digital object identifier or a DOI associated with that static narrative that can be linked and referenced within your publication. And we can also help with some version control if you need to reference or put in your particular publication that's cited this the static narrative, we can help you update the version that's associated with your DOI once you have that update to add. And we have a partnership with the American Society for Microbiology resource announcements to really help streamline publishing. We've gone through our template and our checklist with them, and they've been quite supportive. And we also end up working with them to have a student centric microbiology resource analysis section that I'll show toward the end. So what I like to do now is go into KBase for a bit more of a demo and follow along. So I have 3 different activities that we're going to walk through over about the next 20 minutes or so. And then we will have time for breakouts and getting into to do these activities together. So the first one is a beginner or like a beginner intro to KBase activity itself. I think Lana just shared the narrative with everybody. And let me toggle over here. Okay, and I will toggle to view only mode. Can everybody see the narrative itself? And I know I only have one person who has their camera off. Okay, thank you. Thanks for the thumbs up. Awesome. Okay, so this is an intro to using the narrative KBase activity. This is, you must be logged in to see and work in this particular workflow. You will have a one that is view only. So you can see the different markdown items in here. So all these different markdown cells. This is really just documentation. If you open up, you can also open up the little data toggle or the panel toggle, and you'll see that there's no data in this workflow just yet. And then you'll also see that you can look at an outline of this workflow, and these have all the different markdown cells. So it's an intro to using the narrative. We have some resource shortcuts, how to make a copy, adding data, running an app using Markdown and Share. So if we go down to our making a copy of the narrative, since this is in view only mode, again, over here, we have our data pane. We also have the apps, which is below. So these are all the different applications that are available within KBase. And then up here, we have the name of the narrative. We have my username. This means that you can always look at this particular narrative. Up here in the toolbar, we have a copy button. We also have a help, and we have the share button. So what I'll have everybody do, if you're following along, is you will click on copy. And then I'm going to give it the name Fall Educator, or I guess I can just call it Educators Orientation 2025. And I will make a copy of this. And then I'm going to open the new narrative. And so this is spinning up a new version. And so if you're following along and doing this, you are also spinning up a new version of the narrative that you can work in. Okay, so now I have a workable version, and up here, that little eyeball has now transformed into a pencil. And if I want to, I can toggle into view only mode, but right now I do want to work within this workflow. And I already made a copy, so I can use this table of contents because it has been hyperlinked. So I can go down to my adding data button. So now that I have an editable version following along with the instructions, I can go to the add data button, and I'm going to look at my favorite microbe, and then I will aggregate it to this particular narrative. So adding data, when it opens up the data panel, you have data that is already within your narratives. You can access data that has been shared with you. There's also public data, example data, and import data. We aren't going to go through the import data, but when you click on the import data, this is where you can drag and drop your data. So just files from your computer. You can also access the upload with Globus, upload with a URL. There is a tool that you can access in the apps if you want to upload from SRA directly. So there's an import SRA reads as files from the web. So you can also use this as a way to import data. But for this example, you're going to go to public. And we are recording this. So if I'm going a little fast, I'm sorry. But you'll be able to go back and watch the recording. So in chat, does anybody have a favorite microbe that you would like to look at? I'm going to choose from NCBI RefSeq genomes. There's also microcosm cytosome genomes. We also have RNA-seq reads, some plant RNA-seq mutations, public ontologies, and mycology translations. Some examples. We have one request of AMF, which Jacqueline, can you please write AMF out? Because I'm not going to be able to just type that. Oh, you're making it hard for me. You want to go for fungi. I don't know if I have that one, but I could look in microcosm. Let me see if that is one. Oops. Let me say that. That's on micro. You've stumped me. All right, I'll try it. The next one. There we go. We have two options. I have one that has a single contig of course and two features, and one genome that has many more contigs and also many more features.
Yeah, the second one is the virus of the organism.
Oh, that's a virus.
Yeah.
Thank you. I'll add the first one. So clicking on the add data button, the blue button, I've added data to the data pane, and now that's popping up within my data pane. When I'm done adding my data, I can either click the hide show data browser or this close button. One way that I can also look at my data within the narrative is I can click on the data name and it will populate within my workflow. And then when I'm ready to run an application for it, which I want to run check M, there's a couple different ways I can go to it. One way is to open this data object within the data pane. So that's by clicking on the name. Also by clicking on this little three-dot menu button. And then below the name of this genome, I have a few options. I have a show apps with this as input icon, show apps with this as output. There's exploring the data. I don't have any reports associated yet. There's reverting, viewing the history, examining the data provenance, downloading or exporting, renaming it, and then also removing it. If I want, I'm going to select show apps with this as input. And this is immediately going to start filtering the different applications. There's a few other ways that you can also filter data, or filter tools, either by category search, there's a refresh, and then there's also a way to look at released or beta versions. Released is going to be the default. And I want to add check M below this particular markdown cell. So I have this markdown cell selected, it's highlighted, and it has a little green tab. I'm again going to check under the KBase genomes, and I want to look under genome assembly, and this is where check M is. So if I click on the name of check M, this is how it is going to go into add the tool to my workflow. And you can see when you look at the outline that now we're slowly building out this particular workflow with data and with different tools. So the way I'm going to add data is under the input objects on the configure tab. I will click on the configure tab, and I only have one genome, one data object. If I had other assemblies or bin contigs, they would also show up here. I will select that. I can look at the advanced parameters, but I'm happy to keep these, keep the default parameters. If I'm confused about any of the particular parameters, I can go over to info, and this is where I can look at genome quality, all this other information, information about the parameters themselves, and then I'll click that. And now that this tool is running, I can view the job status. This will tell me if the job is in the queue or how long it was in the queue for. And then it will also give me information for how long it's running. If I run into an issue or want to see what might be going on with the job, I can view the log, and this will give me information on any hiccups or any issues that might be happening with that particular, with this particular job. So that'll be running for a bit. The next item that we're going to look at and kind of go through during this demo is using Markdown. So the Markdown cells themselves are editable. So this is where you can edit and change any scaffolding. I have an example that you can look through here with changing by picking your favorite color. To edit the Markdown itself, you're going to double click on the Markdown, and this is where you can change the text. And I can write in, my favorite color is turquoise. And that's how you edit. There's also more information on changing text code and that sort of thing. We have a Markdown cheat sheet that is also accessible in, I think, the resource shortcuts, where we have, sorry, that was fast. Where underneath the documentation, we have documentation on how to use Markdown, and then we have this narrative Markdown cheat sheet that you can click on. And this particular narrative is just Markdown information. It goes through HTML language and Markdown language and how you can utilize that to change the appearance of your documentation or your guidance for students. So here's our different Markdown guide when it's to view what the formatting is on the back end. Again, you will double click, and then you can see, for heading 1, we use a hashtag space, and then the words that you want to use for that heading 2 hashtags, bold text, 2 start, italicized 1, information on block code, block quotes, and then we also have an HTML, which uses a little different language. So that's where it has like the heading 1, heading 2, and you have those in the brackets. So that is a resource that's available for everybody. It's accessible both through this interactivity as well as included in a few other resources for educators. Let's see. I want to check if my app is still running. Okay, my app is still running. So the next part I have is using teaching workflows and kind of going through the resetting and editing existing workflows. Within the educators org, we have a resource that goes over all of the different workflows that we have in KBase. And this is our overview. So there are 2 versions. If you go to search within the narratives out here, we have a student version and then an educator version. I need to update the student version. So the educator's version is the most recent. And if I click on this, it will open our educator version. If you have not looked at a narrative before, or have not opened a narrative within the org, you might have a click for view access, or you need like a little button that pops up. Click on that, and then once that little pop up, but once that button has gone away, then you can click on the name and we'll be able to open the narratives. If you are in the org, all of these narratives, all these different workflows will be editable or will be viewable by you. And again, you'll see it in the view only mode. And here in ours, I have a nice big tree of all our different workflows. So that's just a reference for different workflows that are available. And then we have the workflows and that are hyperlinked here. So we have the different topics, and we have both English and then some Spanish versions. And the Spanish versions, this is where the markdown has been translated into Spanish. So these are genomics, metagenomics, phylogenetics, metabolic modeling, and genomes, and bacterial RNA-seq modules. There's some information on customizing teaching workflows and editing markdown, deleting data objects, resetting runs, adding new data, and then resources, including a module skeleton, the interactivity that we're going through right now, and then some other resources like our handbook, our educators landing page. So we're going to take a look at the genomics, and we can look at both the Spanish and the English versions. And I'm going to demo a reset and adding data. So you toggle here to view only. So this particular narrative, which they all have a really similar breakdown, where you have KBase in a classroom, genome assembly, modules, there's background context about the particular species that we're working here, Staphylococcus aureus. Information on sequencing is provided within this narrative. Then there's importing data, also importing data. And this particular workflow, since it just goes through the stages of assembly, we have a quality control, reprocessing using Thermomatic, and then reassessing the quality after reprocessing, and then assembling with two different tools. So both IDB-UD and then IDBA-UD, and then spades as our 2 options. And so these are all the different markdown cells and tools that are already within this particular narrative. If I look at the Spanish version, it is going to look very similar, including with images, as well as the data and the outline. The only difference is all the markdown language is in Spanish. So let me go up to the top. So this is super handy to use the outline if you have a long narrative, because you can just click on the different cells and it will take you right there within your workflow. So this particular module has this genome assembly is also linked to genome annotation, genome analysis, comparative genomics. So all of these all these narratives that are part of the larger workflow are accessible here. I'll go through and load. And this is where these different narratives, so the full workflow, are daisy-chained together. So the assembly that was chosen from part 1 has been imported into this particular narrative. So say you want to use this particular workflow with your students, but you would like to have them do their own assembly or annotation. So if you have a longer class period where your students are able to, you have the time for them to either do this during their homework or run the tools themselves, you are able to both delete the data and reset the tools. You would do that by making your own copy. So I'm going to make a copy of this particular narrative and I'll open it. And I want to keep the data in here. So I want to keep these original SRA reads, but I would like my students to do both the trimming and as well as go through the assembly itself. So I'm going to go through and delete my different data objects, which I can do directly here. And so then if I go down to my outline, I will also go through and I will reset these different apps. And I just need to change those. And I can go here as well. I will click on my reset. Okay. And it's reset. It's reset the different tool. So then the students will have to go through, they'll have to go and select the data, go through the parameters, and then run the tool themselves. So that is one way that you can utilize these particular workflows. Based on timing with your students, you can reset particular tools. Just remember that what the tool needs, the data that the tool needs to run, if you're not going to have that already ran, it should be in the data pane so that the students will have access to that particular data object. Again, I mentioned kind of importing data from other narratives. If, for example, with this this particular genome, if I have, this one already has the spades assembly imported, but this can be imported from the from that initial narrative, and you can go through this. And I have so many narratives. You can select the particular narrative that you are importing from. You can also search through these different narratives. So I could say genome. You can search or if you know the name of it, directly just put the name into the search bar. And that would, so I have a few different copies of this because I've made multiple copies of different narratives. But you just want to make sure that you have the same narrative name, and this is where you would copy and add that particular data object to this workflow. And then the other option is to start from scratch without any data And that you can do using the module skeleton. Let me find my overview. And this is where we have a particular module skeleton. And this is really an outline that's based on the narrative outline that we used with each of these different teaching workflows. So again, it's going to be just markdown cells. And we can take a look at the outline where we've included the markdown guide. And then, you want some scientific background. You'll have the module information, and I need to update that. I'm sorry. That should be a little image, but something changed with how the Google imaging or any images I've imported from or been used in my Google Drive changed. So then we have a table of contents. For example, here, this could be the workflow that's just within this particular narrative. You can also link to other narratives that you might have your students go to next. The structure here is providing any sample information, how to add data. If you haven't added data, going through any data exploration, including your applications, results, putting in questions for the students to answer. Again, you can use the Markdown guide to help with highlighting and calling out any different questions that you want your students to answer in their own version and copy of the workflow. And then you can daisy chain these narratives together if you want to have another workflow that they move into. So this is an example of, you can use this as a reference or make a copy of this particular workflow and then start to add your own data, add your own markdown to these different examples. So you both have the option of starting from scratch, such as here, you can use these different workflows that are really built out, and then we also and we have a lot of different workflows in here, both in Spanish and in English, and some examples that have been shared by other educators. Okay, so I'm going to pause the recording and okay, so for those of you who are sticking on for our program updates from this past year and what's been happening with KBase, we have a few items to talk about. The first is students and publishing. So we've been working with the American Society for Microbiology resource announcements, and we have a special collection with them, which is called Promoting Microbiome Workforce Development with KBase. And this is really highlighting students who have been working in their courses to go through the process of publishing new data. We have both isolate and metagenome assembled genomes in this collection. And we're trying to help instructors leverage the use of persistent identifiers as they're helping their students build their CVs. So we recommend students having ORCIDs, which is the Open Researcher and Contributor Identifier account, and allowing us to help you get a digital object identifier associated with the narrative that you are publishing as well. And so we've been going through the best practices for that. And one particular example we have from Anna McLuhan's class. Here they went through and isolated 6 different endospore-forming bacilli species. And so here in this example, the little top clip is from the MRA itself. And then right below that is the KBase narrative that is cited by this MRA publication. So it's included as a data resource. And then we also have the Siena College 2022 MRA genome announcement that also links back to the publication itself. And so there are 16 different authors on this particular example from the class. And there are 6 isolate narratives that are linked from the single parent narrative that Anna put together. The parent narrative is the one that has the digital object identifier, and we are able to keep that workspace number associated with the DOI. So the top URL under the 6 isolate ones, we've included that workspace, so the narrative identification, and have included that as part of the DOI. And so you can actually put in a request to reserve that beforehand. And this particular data was collected by these students, so they went out and they did the collection and the sequencing. We are also able within KBase to track the views, this particular narratives, and if people are using the data for their own analysis. So if there have been any new narratives created from the parent narrative. So that's a cool way to be able to track impact for both instructors and for their students. And so we have the particular promoting microbiome workforce development with KBase. We have the collection. That is the link to it. And then the collection itself, we have different features. So we have a little bit of information about the collection on the page, and then you can access all of the narratives that we have added. And so these are narratives that have student authors. So students have been going through and either doing the lab work and or the data analysis within KBase. So we are highlighting students who are working with KBase and or in KBase to do their data analysis. And this is all kind of part of our bigger initiative within workforce development of microbiome sciences. So in the last 2 years, we've had this microbiome workforce development program pilot that was funded by the National Science Foundation. It was a research coordination network for undergraduate biology education incubator award. During this pilot, we went through and we started developing some modules or kind of modularized the research workflow and started developing teaching resources based around that. And really our goal was to think about how this program could be integrated at any different institution and really used by instructors to meet the needs of their students and really think about how they can help their students contributing to challenges in microbiome science. We have a few different standardized and also how they can generate their own standardized microbiome data sets across programs, make those available through ASM NRA, and ensuring that students receive credit for those data contributions. So again, going back to linking those digital object identifiers and how we can leverage those resources. That has now grown into MICROnet. MICROnet is the Microbiomes and Computational Research Opportunities Network. So this is now a full research coordination network for undergraduate biology education. And this again builds on the incubator, so our pilot from the original educators program, which was centered around data analysis and also includes our publishing efforts. Now we have developed this modularized research workflow that takes that provides resources for instructors to kind of take their students through research question and hypothesis development into experimental design and sample metadata, sample collection and processing, and then tying that back together to the data analysis and publishing. And so the goals behind MICROnet are really to distribute these different teaching modules and expand the educator network. We're also working to establish regional collaborative hubs. This is really to connect different peer mentors, so collect educators with each other, help connect students, as well as provide means for research sharing, as well as instructor training. And then we are also evaluating the program for its use, its implementation, and accessibility. And really our end goal is to kind of achieve a sustainable, long-lasting program to curate a student-curated fair data set, which goes back to our first goals from the pilot. And so with this funding, we're really thinking about how we can provide instructor training, establishing these regional hubs, sharing resources, and developing a sustainable program and network. And you can think about these different modules and how they're integrated as a choose your own adventure. So there's a lot of different options for going in, either from the research question hypothesis side, whether or not you are going through experiment design and stable metadata. We're really thinking about overcoming any questions or challenges around what is environmental metadata? Why is that important to curating these fair data sets that can be reused and cited again by other courses, researchers, educators, and how these can contribute. So we're thinking about how the data can live beyond just your students and your courses, so that other students and researchers would be able to cite and use that data after your students have finished their courses. And making sure that that data is, again, citable and referenced, and that everyone who contributes is able to receive recognition for that contribution. So there's a lot that goes back into the architecture, but you don't have to include every single module here. It's really a choose your own adventure and thinking about how they can fit in with your particular courses. So if you are interested in MICROnet, from the KBase Educators page, you can go to the MICROnet page. And we have some information on the program itself. You can let us know by filling out a Google form if you are interested in participating. And we also have these resources that are accessible outside of the program. So these are open access resources that we're still developing and iterating on, but we would love your feedback. And again, if you're interested in joining a hub in one of our upcoming years, we just started our first one in the Northeast, you are welcome to let us know. So in summary, we have gone through this fall orientation. We've introduced the KBase Educators program, shown you where to find the different educator resources. We've talked a bit about implementing the teaching workflows and some best practices, both through the Educator's Handbook, as well as how to use the teaching narratives, whether you are customizing them for yourselves or developing totally new ones, and gone through some of our newer initiatives with MICROnet and our ASM NRA collection. Different resources for you and next steps. Again, we have our KBase landing page, documentation at docs.kbase.us. Our YouTube channel, LinkedIn is a great way to find us, and more places about how to contact KBase itself and find resources that you need. And again, if you are interested in KBase Educators and MICROnet, whether you haven't been too involved or want to get more involved, you can look at our landing pages, and you can also contact me at egdaff@lbl.gov. And that concludes our webinar. Stop recording.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.